I am glad that many of you could make it, and so we're going to get started here in about 4 minutes. We want to be punctual because I want to respect everybody's time. Thank you all for joining, and I'll come back in about 3 minutes to touch base, and then we'll get started. If you are joining us and you want to head over to YouTube, you can do that by clicking on the YouTube button on the presentation page. Then you'll be able to answer any or ask any questions you like. I'm going to head over there to make sure that I keep an eye on anyone who's joining us for questions. And we look forward to talking through a great presentation on the microbiome, but we'd love to hear your questions specifically so that we can respond to them and effectively give you clarity on what the microbiome is all about. So we'll get started here in about 2 minutes. For those of you just coming on, we're going to start in about 2 minutes, and we want to give everybody a chance to jump on. If you want to click on the YouTube button, we welcome your questions. We are going to hear a great presentation on the microbiome beyond probiotics. Of course, you're welcome to go back and watch the previous webinars that you'll see on the presentation page. But I hope you'll enjoy this one, and feel free to ask questions that you might have regarding the microbiome, testing of the microbiome, and concepts that we are going to be touching on in the in this presentation here. All right, it is now 4 o'clock, so I want to welcome everyone currently on, and I know that we'll have others joining. My name is Francisco Rodriguez, and I am the founder of Supplement Innovator, and we are joining you in West Palm Beach, Florida, and our presenter is joining us all the way in San Francisco. I'm really excited about you all participating with us on this innovative topic regarding the microbiome. A little bit about Supplement Innovator. We are a consulting firm to the nutritional supplement industry. Particularly, we focus on training ingredient sales teams regarding how to effectively add value to the entire value chain. And then there's another area that we cover that is part of our innovation story, where we bring novel ideas to the industry that allows them to maximize those ideas similar to the topic we're covering here on the microbiome. And I want to invite you all to ask questions. If you are on the presentations page, supplementinnovator.com/presentations, you can click on the YouTube button on the bottom right of the video screen and head over to YouTube and ask some questions regarding this presentation. Maybe you have questions on a previous presentation that we've done that you'd like an answer to, and we would love to answer those questions for you. Our guest today is Second Genome, and we're particularly joined by Second Genome's Take Ogawa. A little bit about Second Genome: they are a company that brings microbiome science to the discovery and development of therapeutic products. The company has established a pipeline of microbiome modulators that impact infection, immunity, and metabolic disease. Second Genome's pipeline is fueled by novel technologies for identifying, screening, and scientifically validating product candidates and microbial biomarkers. Second Genome's technology have been rigorously validated through partnerships with leading pharmaceutical companies and nutrition companies, as well as academic and governmental institutions. Some of their work you can see on their recent news, and they have a great relationship with major pharmaceutical companies. And we are grateful to enjoy the presence of Take Ogawa. Take is the Director of Research Services at Second Genome. He spent the last 15 years in the life sciences industry, developing and promoting genomics tools and applications. Take was part of the development team that launched the first high-throughput sequencer at Applied Biosystems that was responsible for sequencing the first human genome. He also promoted the first next-generation sequencing platform at 454 Roche. According to Take, he has seen the industry shift the research focus from looking at just changes in our human genome to also looking into how we interact with our environment in and around us. And Take is going to be presenting to us today's webinar, which is called Beyond Probiotics, and we're going to be discussing how to set up your own clinical study. Take, welcome.
Thank you, Francisco. Appreciate the warm introduction. So
I'm going to give you screen sharing.
Great.
In just a moment.
Thank you. Great. Thank you, Francisco. So let me just pull up my presentation here and get it in presenter mode. All right. Again, thank you, Francisco, for the introduction. So, as Francisco mentioned, we'll be covering two broad topics today, leveraging or really riding the coattails on the previous talks that we've had as part of this series, and so this is the last of that series, and we'll be covering two topics today. The first topic is looking at the microbiome beyond probiotics, and then finally wrapping up with discussing and sharing ideas on how to set up your own clinical studies in the context of looking at the microbiome. So first, to address the microbiome beyond probiotics. So just to get everybody on the same page here, when we think about the microbiome and how we define it, really we're talking about this in the context of microbial communities that take residence within the various ecosystems, if you will, within the body. And of course, there are many different microbial communities that exist. Excuse me, but specifically, we're talking about in the context of the human body and human health today. So as you can see from this picture, there are quite a few different communities that take up residence in our host, and you can see that depending on where we see, the communities can vary quite differently between the gut, the oral cavity, our skin, and even different parts of our skin between our hands, our feet, and so on. And so this has been known for some time that these different communities exist, but what has not been known is, what has not been available, rather, technology and the knowledge base to study these communities and thus make sense of how these communities are shifting and their function and purpose. And so that is what we're learning today, and that's what we've been discussing in this webinar series. So more specifically, if we focus particularly on the gut microbiome, so this is the bacterial communities and other microorganisms that take in the gut in particular, we see that we are finding that they play numerous critical roles and functions within the well-being and the biology of the human host. But specifically, when we look at the gut microbiome's function in metabolism, we find that those particular functions are quite extensive. The bacteria are playing many different roles, such as synthesis of vitamins, fermentation of non-digestible fibers, and other supplements and diet. And so specifically today, we'll be talking about some of these concepts around how supplements and dietary components that we consume plays a role in our health, and also how the microbiome is an integral part of that formula. So specifically, there are studies that show that highlight evidence that certain types of fiber, dietary fiber that is, that we consume either independently as a supplement or that are part of our diet, play a critical role, or metabolize either directly by our systems, the human GI system, that is, versus how they are metabolized by bacteria that take residence in our GI system, and thus produce various byproducts, metabolites, or some may call bioactives, that have been shown to play an active role in our health. And these studies have been replicated in animal studies as well as in clinical studies in humans, and particularly fibers such as inulin has been getting a lot of focus in its role in its role in terms of its direct impact on human health, and also effects on different clinical outcomes such as weight loss and so on. So so there are many studies that highlight this, and here is just several of those that we find of particular interest that have been recently published in the last couple of years, really, where again finding direct correlations between the fiber itself, how it's modulated or metabolized by certain strains of bacteria, and how through that various bioactives such as short-chain fatty acids are produced, and how those metabolites have a direct impact in our health. Various studies, and these are just again a subset examples of such studies, peer-reviewed studies that highlight this very important evidence. And when we look to further mechanism of how the microbiome ties into, again, using the example of dietary fiber, and how those fibers then are metabolized and consumed by the bacteria, thus then the bacteria will generate certain bioactives such as short-chain fatty acids. And here in this picture, we see that those short-chain fatty acids such as propionate and butyrate and such play a direct role in certain functional pathways within our own gut biology, in this case specifically around inflammation, in that these increase in these short-chain fatty acids have shown a decrease in inflammation in the gut. And so really ties the story together nicely between the fiber supplementation and its role directly to the bacteria, and then how the bacteria then interacts through these metabolites and fatty acids with the host system. So we've completed the circle, as it were, in terms of how this all ties together, understanding each step. And these studies are possible today. Now, of course, there are many other types of supplementations to consider and other components of our diet to consider. And this, again, is just highlighting some of those examples. So going beyond fiber, there are things such as polyphenols, specific vitamins. So here's a vitamin D story that was recently published. Yet another vitamin D story in conjunction with probiotics, and its correlation or association with certain clinical allergies, asthma, and obesity. And then there's also, counter to the beneficial effects of such supplementation, there are also stories where it's the exact opposite. So there's a very well-referenced, well-cited study of artificial sweeteners in that it induces glucose intolerance in animal studies and human studies, and how that ties in with the gut microbiome. So it can really go both ways, but we're able to characterize how certain supplementation, how certain dietary components can either, that can influence the gut microbiome in either a positive or negative way. So moving beyond this, getting into the second part of the topic that we wanted to discuss, considering through the series of these webinars that we've been completing, I've depicted and shown using real evidence and real science here that that there's very strong, there's a lot of opportunities here for each of us as we consider various product development or marketing strategies about our products, about how we can go in and set up our own studies to build this sort of knowledge base and build this, further understand how exactly are our products influencing or engaging in a specific health outcome. So I want to go into a little more detail about how that is enabled today with partnerships with companies like Second Genome. So specifically, when we talk about doing conducting a microbiome analysis, there are really three major components to consider at a high level. One is, of course, which samples do we analyze, and how do we collect those samples? How do we store those samples? The whole logistics around to collect and measure is a key part of a key consideration in setting up your own microbiome study. The other component, major component to consider is, well then, once those samples are collected, how do we actually measure the microbiome? So now we're getting into the details of which technologies, which laboratory methods do we use to accurately and reproducibly measure the microbial community. Excuse me. And then once those measurements are made, we are collecting a vast amounts of raw data. As we all know, the microbiome is made up of, or depending on which microbiome you look at, but specifically around the gut microbiome, we're talking about communities that are made up of hundreds, if not thousands, of different organisms. And those organisms, so to accurately measure, identify and measure that microbial community, we need, and we require very strong bioinformatics capabilities and know-how to interpret those results and build certain insights and answer certain questions of that data set. So putting these three major components together in a, again, robust, accurate, scalable way allows us to gain further insights, and those insights is what translates to building better products and also providing better marketing knowledge as to how your product is differentiated in the marketplace. Critical considered in parallel ways. And so, second, as Francisco mentioned, we have our own drug discovery R&D program, where specifically looking to develop therapeutics in the realm of, particularly right now, we're focused in the gut health area, and some of the examples that Francisco mentioned are those efforts to do our own R&D in partnership with pharmaceutical companies, all in the hopes of finding us developing drugs. In parallel to that, Second Genome services side of the business, which is where I sit, where we actually cater to projects and studies for our external clients as a service. And so in that context, what we're doing is taking on, enabling our clients to set up their own experiments, to collect their own samples in the context of their study. Those samples are then submitted to us. We prepare those samples, we analyze those samples, and on the back end provide a publication quality report that allows our clients to then consider, again, further experimentation, publication, product development initiatives, follow-up studies, and building marketing literature and such. So it is a end-to-end solution that we provide here for our clients that are interested in doing their own microbiome study. And in that experience, really, we've been doing this since early 2010, is that we've actually processed over 150,000 samples of various sorts and various projects. These are all, again, external projects that we've taken on for the benefit of our clients. So we've accumulated vast amounts of knowledge and experience from processing all these samples. And just to put this into perspective, one of the largest well-known human microbiome studies that are out there today, which is the NIH study, has only processed well, essentially about a third of the number of samples and analyzed about a third of the samples that we've processed. And you can see that, in fact, we're really on an exponential learning curve here in terms of the amounts that we're gaining from these studies, which, of course, the learnings from those studies, rather than the insights, of course, can be shared across these studies in terms of our expertise in processing samples, analyzing the results, and so on. And really, though we have a focus on the human health sector, we do cater to many different disease areas or therapeutic areas and applications within the context of human health, and we've done over 300 studies in that context. And though we've been mainly focusing on the gut microbiome, which is often sampled through stool samples, there are other, of course, many, as previously discussed, there are many other microbiomes that take a residence within the human body, such as in our mouth and skin and so on. And so you can imagine that there are many other types of samples, depending on the questions at hand. And we've done a wide array, we've processed a wide array of samples with reproducible results. Now, when we talk about analyzing a microbiome, that's actually not, there are many ways to look at the microbiome, I should say. There are many lenses we can use to start to identify microbial communities, but there are also questions around function and activity, and how it ties into other aspects of biology. And so this is just a simple depiction starting from the top of the various applications that Second Genome offers, which allows our clients to look at the microbiome in many different ways. And so to summarize here, each of these applications and techniques have their own strengths and weaknesses in that they all generate different types of data. But all these data can be integrated to one another to offer additional insights. So starting from the top, what we call 16S sequencing, or rather 16S analysis, is really looking at the 16S gene that is present within all bacteria. And 16S, that particular gene is essentially the fingerprint of all these bacteria that allows us to identify who's present within a community. And then we can also measure the abundance of those bacteria based on the abundance of that gene within a sample, and thus we can measure relative abundance and community shifts from sample to sample. So we can measure how the microbiome is changing based on a study. And then beyond that, of course, we want to start to understand if there is a shift, then what is the function of that shift? What are the certain pathways that might be affected by the change in those communities in terms of a metabolic pathway or an inflammation pathway and so on. So there are different applications and technologies that we use, such as metagenomics, metatranscriptomics, to, again, start to elucidate the function of the and what those shifts actually mean in terms of the biological impact. And then going beyond that, of course, looking at the metabolites that are released by these bacteria, which we call metametabolomics, and proteomics, which is to look at the proteins that are secreted by these bacteria, is yet another application of looking at what you could call the business end of the bacteria, and which is the part of the, is again, as described earlier, is what interacts with the host. And so to understand all of these different aspects of the microbial community allows us to gain further insights into the mechanism and the function and what all this change means. So when we start looking at the data, and I don't want to spend too much time here because this is, again, more specific on a case-by-case basis, but just to give you some taste for the kinds of analyses we can do and the kinds of questions we can ask, which is rather important. Of course, as I said, when we do community profiling, really the kinds of questions we're asking is, who is there, their relative abundance to one another in terms of species A in sample 1 versus species A and sample B, and how are they changing, or species A and species B within a sample can also be measured. And then most importantly, what we want to understand is how is that community within, say, an individual changing from time point to time point, or say treatment to treatment. So we can measure that based on this sort of application. And then again, going beyond that, this is what we call a heat map analysis, where again, we're comparing a group of diseased individuals versus a healthy individuals. And you can see based on this heat map analysis that the left side and the right side of this heat map in the middle here is different, and that's because they're sorted by diseased individuals and healthy individuals, and the rows represent different metabolic pathways. And so you can see that it's very different. And so these sort of analyses, allows us to start asking questions such as, within these microbial communities, which genes are present, how are they being expressed, which pathways do these genes represent, and what is the function or the functional change based on how these genes might be shifting. So we can ask a lot of questions from a given sample that's collected in a study. So to highlight again where we fit into the workflow of setting up this sort of study of your own is everything you see in blue is what Second Genome does today, and what we can offer. What we are not currently doing, at least within the walls of Second Genome, is the sample collection piece, which includes steps such as patient recruitment, patient questionnaires, sample collection, dosage of the particular product, and distribution of those products, and so on. That is typically done by the clients themselves, or through a CRO, which is essentially a contract research organization, which we have several existing relationships with. But essentially, through partnerships, the sample collection piece is managed, which of course is an important step in the entire process. And so in that, as I said, the logistical piece of that is we work with partner laboratories for the recruitment of the volunteers for a particular clinical study or a clinical trial. They also manage certain data collection pieces of that in terms of whatever we may be interested in measuring, such as body weight, insulin sensitivity, or insulin sensitivity, and so on, and of course collection of the samples. And then once those samples are collected, those are delivered directly to us for processing. So we are continue to invest in minimizing the number of steps and simplifying these clinical trials. But needless to say, we have already engaged in a number of these studies and have seen really good results and quick turnaround time. So just to paint a picture of a typical study that we do see, in this case, what we are representing here is a microbiome study to look at the effects of inulin supplementation in overweight patients, or not patients, but in this case, participants or volunteers. So in this study, what we're doing is we took 50 volunteers, all with a BMI greater than 30. We're going to split that group into half and put one arm on the inulin supplementation and the other arm on a placebo supplementation. And from each of these individuals, we'll collect a stool sample prior to starting the study, so to build a baseline. And then these individuals will be on their particular diet supplementation for a 2-week period. And then at the end of the treatment, as it were, we will collect another stool sample from each of these individuals. So this is the basic study design that we can imagine. And then what this sort of study design allows us to ask is highlighted here, which is again, the basic questions that we can ask is how are those microbial communities changing from pre and post the intervention of the particular dietary component, in this case, again, inulin. We can look at how the individuals that were on the treatment differ from the individuals that were on the placebo treatment. We can also measure short-chain fatty acids within the stool to see how the short-chain fatty acid concentrations are changing, and how does that change in concentration correlate with specific changes in the microbiome or specific organisms within that microbial community, and build the statistics and the correlation around that. And we can also measure comparisons between changes in the microbiome to weight. We can also filter by gender, and we can also filter this by insulin sensitivity, which will be measured in parallel as well. So the inclusion of these various, what we call metadata, such as weight, sensitivity, short-chain fatty acids, is an integral part of our offering to gain the most insights as to what is happening in the microbiome. And then these sort of insights in our report will allow consideration of further studies in terms of new questions that come up. And that's an integral part of our service as well, is the interpretation of those results for consideration of new questions or new hypotheses. I'll go ahead and stop there. And Francisco, I'll hand this back to you for a question and answer session.
Take, thank you so much for that presentation. I have a particular question. I'm just checking to make sure that nobody else does yet. So my question is specifically around a point you made in slide number 14. I have the privilege of having the slides available. And what's exciting here is that the data that you're extracting isn't particularly focused on health outcome, but it does tell a good story. So can you go through, it seems like there's identification, functionality, and then we have two others that are a little bit downstream from that. Can you just go in a little bit of detail about those steps again, so that we can get some clarity around the results that most people would get to see?
Absolutely. So with everybody out there that's doing the microbiome, the basic that is researching and analyzing and studying the microbiome or have interest in the microbiome, for the most part, starts out with is, I believe there is a microbiome role, an important microbiome role in my study. It could be a diet study, it could be a supplement study, it could be a therapeutic study. Whatever that study is, there's a general basic hypothesis that I believe the microbiome plays a role. And to be able to answer that question, or to start to answer that question, the first thing we need to do is, one, identify the members of that microbiome. So the microbiome, again, in this context, is a microbial community, specifically a bacterial community. And we want to first identify, without any prior knowledge, who those members might be. What are those strains? What are those species? What is the genus of those organisms within a sample? To answer that question, there's essentially one technology that allows us to do that today, which is, as I mentioned, it's what we call 16S analysis, which is a gene that is that exists in all bacteria, but the sequence of that gene, the DNA sequence of that gene, differs depending on the organism. So essentially, depending on the strain of the organism, that is to say that it is its fingerprint. Just as different people, different humans can be identified with their fingerprint, it's the same exact thing here, where if we can sequence that particular 16S gene in all the different organisms that reside within that community, which is exactly what we're doing. We're literally sequencing those genes or probing those genes using genomic technologies. We can identify that sequence and thus identify which organism that particular sequence came from. And not only that, these technologies allow us to be somewhat in that if there's more copies or more representation of a particular type of organism, then we'll actually collect more reads or more counts of that particular organism, so we can start to build a relative abundance profile within a community to say that bug A is twice as abundant as bug B, which is twice as abundant as bug C, and so on. Because having that relative abundance information is quite cool. And the most important question that we want to ask in this context is not so much who's there and how many of them are there, but rather how based on your treatment or based of the subject, that is. So are they on a certain drug? Are they on a certain probiotic? Are they on a certain diet? Are they on vitamins and so on? To measure that shift tells us that it starts to allow us to imply, associate, that is, that your particular product, your particular treatment is impacting the microbiome. So we can start to answer those questions. And then naturally, once we know that the microbiome is shifting and changing because it is being impacted, well, we want to know what that impact means. Well, 16S analysis does not necessarily allow us to characterize what that impact means. The only way to understand what that impact means, what that shift means rather, is actually to sequence the entire genome of every bacteria within a community. And to do that is to sequence the entire genome. And the way we do that is yet a different kind of sequencing technology. But once we can sequence, we can thus identify the genes that make up these organisms. And the genes tell us the function of these organisms. And we can start to measure how those function and overlay those functions on a particular biological pathway, such as a metabolic pathway. And thus, we can start to see and actually decipher how the function is changing the community or sample to sample based on a particular treatment. So metagenomics and metatranscriptomics offers us similar inputs. One is looking at DNA and one's looking at RNA, but I don't want to spend too much time on those particular nuances. But the idea there is that we want to start to elucidate function, and that's the same idea with metabolomics. Now, rather than looking at the genomic information, we're now looking at the proteomic or the metabolomic information and correlating the two data sets to make sense of the function of the particular community.
No, that's very helpful. And I want to clarify for everybody, that allows you to tell multiple stories as a brand or as an ingredient supplier about your product. And so I had Take go through that because I think that was a question that I'm getting a lot about what the data show. We have three more questions from the attendees. Number one, does Second Genome quantitatively analyze short-chain fatty acids in feces in-house, and by what methodology? And the second question is the analysis of bacteria present in samples qualitative or quantitative? And I think you answered both of those before, but I think it's good to hit them again.
Yeah, absolutely, great question. So yes, we do offer measurement of short-chain fatty acids through our services, because again, we find that to be a valuable piece of gaining insights from the microbiome, because again, most short-chain fatty acids are direct byproducts of bacterial fermentation. And we actually enable various technologies. We have several options in terms of how we measure short-chain fatty acids, and also that's defined by which particular short-chain fatty acids you're looking to measure. But you can some of the basic technologies, of course, is GC, gas chromatography, and we're talking mass spec and various technologies of that flavor that we can use for these sort of analyses. But particularly, I think it's important to keep in mind that because we're talking about stool samples, that there's a unique handling and care that's required for those sort of samples in terms of not only the logistics, but in terms of how we measure things such as short chain fatty acids, which again we have experience in. And so just to put that out there, that is an important consideration when thinking about how these are measured. And so again, we have experience around specifically in working with stool samples. And then to the second question, exactly as you said, Francisco, is that yes, these technologies are quantitative in a relative fashion. So we're not measuring specific cell counts per se of a particular organism. We can surely integrate those sort of methodologies and readouts using other technologies. But really, the goal of these technologies and how they've been optimized is rather to look at the community at large in a de novo way, and thus measure relative abundance of each organism to one another. So I'd call it is a semi-quantitative technique at this point.
And I think that just to reiterate that point, I think any technology is not fully quantitative as far as all of the bacteria are concerned, because it's not necessarily possible to measure some of the organisms inside of the gut, because they don't grow in environments that we've been able to create necessarily in the lab setting. So I think those are some issues that may be something to consider.
Absolutely, Francisco. I think historically, when people think about microbiology and bacterial analysis, we often think of culturing assays where we're counting colonies in a culture or in a petri dish, and in those cases we're limited to the number of organisms that we can interrogate to basically, as you said, only organisms that we can actually grow in culture. Rather, what we're talking about here, and really the precedence for why the microbiome space has come to be, is in the advent of technology such as DNA sequencing, microarray, and so on, that allows us to look at a sample with no prior knowledge of who may or may not be in that particular community. And of course, DNA is DNA. Everything culturable or unculturable has DNA. And we know that most organisms are in fact unculturable. And these sort of studies have shown that, in fact, relative to the kinds of organisms that have been cultured, say from a gut sample, that when we actually do these microbiome analyses, that we're finding far more diversity based on a genomic approach. As I said, hundreds, if not thousands, of different organisms are being captured using these techniques relative to, say, a few dozen using culturing methods. So absolutely, it's a total paradigm shift.
So we have one more question that's a little bit focused, but it's one that I like because Take is going to be a father for a third time, and I'll be a father in about 9 months for a second and third time. We're having twins. So the question is, has Second Genome done any particular work on the ID and the impact of breast milk organisms on infant health?
The short answer is yes. In fact, one of our clients, and I'm blanking on the name now of the paper and the academic institution that conducted this study in collaboration with us, or through us, I should say. But that is a very hot area in the microbiome space, as you can imagine. Is, in fact, where does the microbiome come from time point zero, if you will, of a newborn's life on this planet. And there's a lot of data, a lot of studies showing the differences in the microbiome signatures between C-section and natural birth babies and so on, and of course breast milk versus formula. And so what we're finding there is that yes, natural ways, a natural birth, natural in the sense of breastfeeding and so on, seems to offer an advantage in the context of the microbiome. But we also know that the microbiome, the gut microbiome in particular, and I can imagine most microbiomes, within the first 3 years of life is very dynamic, and it's very vulnerable in that sense. And so it is an area of deep research in terms of not only how do we feed our babies and infants, but also what kind of drugs do we put them on? As we know, at least in this country and many other modern countries, antibiotics is a major it's a major concern these days, and for legitimate reasons, I would say. And so yes, these areas are hot topics, and thus yes, Second Genome as a whole have seen a lot of these studies in all those applications and all those storylines that I just described. We've seen numerous examples of each of those come through our doors.
That's
so,
fantastic.
Francisco, I'll send you that paper when we get off the webinar here, and you can share that citation as you see fit.
Fantastic, Taki. That sounds great. So, a little bit more detail. I don't know if you have done identical twins, that one has been breastfed versus one formula fed, and determine some of the downstream effects. But that's kind of the detail of the answer that our questioner has asked, and it's a good question. I just don't know if you've had a chance to dig into that.
There has been quite a few. So as for very good reasons, twins are great models for various scientific studies, particularly these sort of studies where we're looking for environmental factors. I think we can all say that the microbiome is an environmental factor in the sense that it's not part of our own genetic makeup, and that it is very dynamic and can be influenced by environmental stimuli. So in that sense, twins make a great model because they're genetically, assuming these are identical twins, of course, make great models for that. And that's exactly why a lot of groups start off with animal models, because you can imagine with animal models, we are talking about knockout models that have genetically identical makeups, and such. We can essentially eliminate that variable from the equation. So when we see different impacts between different individuals that are twins, then we can start to say, well, at least we know it's not the genetics, that it is some other factor that's contributing to the differences. So Second Genome as a whole have not particularly tackled twin studies, but there are a lot of good twin studies out there in the literature that are highly cited for those purposes, that clearly start to show and highlight mechanism of the microbiome that is purely microbial and has nothing to do with or minimal impact by the host genetics. So those studies are out there.
Take, thank you so much for your generosity on time. Thank you for the great presentation. And I look forward to continuing to discuss and dialogue about this. And for everybody that was watching, or for everyone that will forward this on to colleagues, customers, and others, I believe the microbiome is the story of the nutritional supplement industry. It is the one whereby we provide the body and all of its moving parts, which include the microbiome, with exactly what it needs to maximize human health. So I hope that you'll talk to your teams, talk to your R&D teams especially, and feel free to get in touch with me, and I would love to connect you with Take and Second Genome on work that can be done. And I know this seems like a lot of work and a lot of research, but Second Genome has already started some great research in the nutritional supplement industry. So I hope more will be joining those projects. Take, thank you again. And for everyone else, we'll catch you on the next webinar.